Hi everyone, welcome to A&P 2. I'm your instructor Zach Stelter, and you are in for a treat this semester. This is one of my favorite classes to teach. I very much enjoy studying how the body functions, and there's a lot of rich physiology in this semester of A&P. We start with the blood, and the blood is a great place to start because it stands kind of central to what's happening in the physiological landscape of our bodies. There's an interesting picture that I like to reference that shows a cartoon of a cell, and this cell represents, nearly every cell in your body. Most all cells in your body have a couple common features to them. One, they produce CO2. This is what happens when they break down energy molecules. The carbon in those energy molecules and some of the oxygen is released as this gas called CO2. Also, when cells make energy, oftentimes they utilize oxygen. Oxygen is used as the final electron acceptor to take those electrons coming off the electron transport chain. Okay, the cells will require nutrients, so these are things, like energy molecules, vitamins and electrolytes, and any other thing that is required for a cell to sustain its life. And then in turn, there are other wastes and things that the cells will get rid of. In essence, the story of your life is all about keeping the cells in your body happy. And you have many, many, many cells in your body to keep happy, right? You have several trillion cells in your body that all have to stay alive. And the way that they stay alive is by maintaining these exchanges of things all of the time. Well, the way that those things are delivered to and from the cell is through the highway of the blood. So here this represents a blood vessel, and oxygen comes into the blood and then it's brought throughout the body and delivered to the cells. CO2 that's removed from the cells goes into the blood and then it's brought to the lungs where then it's removed. All the nutrients that you absorb from your food through your digestive system, those will come into the blood first, and then the blood will then bring them all out to the different cells in your body. And then if your tissues have any waste, the waste go into the blood, and then they get filtered into the urinary system and expelled. So you can see this is a major medium by which things will travel, and the blood is what's going to be kind of the carrier that brings all of the cells what they need and takes away from the cells everything that they don't need. This class is really a topic about blood, how we get oxygen into the blood, how we get CO2 out of the blood, how we get nutrients into the blood, how we remove waste. And along the way, we'll talk about all of these systems. We'll talk about the respiratory system, we'll talk about the digestive system, we'll talk about the urinary system. We'll also talk about in this class other systems like the reproductive system, the lymphatic system. That's how we keep our bodies healthy. And the endocrine system, which is how we send communication signals through the blood. So lots of good systems to talk about. All right, so above and beyond just being kind of a carrier of important things to and from cells, there's a couple other functions of the blood. The first of which I've just mentioned, that it transports stuff. So it transports dissolved gases, nutrients, hormones, and metabolic waste. But there's a couple others. It's going to help regulate the pH of the body. It does this because there are these things called buffers in it, and a buffer helps to stabilize a pH. If there's too much of an acidic compound, a buffer can be utilized to transfer that acidic compound into a temporary substance, which can then possibly actually get removed as a gas through the respiratory system, or vice versa. We'll talk more about how this is done when we get to the respiratory chapter. But for now, you can just know that it helps regulate our pH. It also helps regulate our ion composition of the interstitial fluids. Outside of the cells of your body is what's called the interstitial fluid. This word literally means the fluid that is in between the cells. And this is kind of an interesting fluid because it has the same composition roughly anywhere that you test it in the body. And that's maintained by the blood taking extra things out of the fluid or putting things into the fluid depending on the needs. There's a very specific amount of things that have to be in this fluid surrounding the cells in order for the cells to be happy and to work properly. And the blood is going to be the thing that helps to maintain that consistency of the composition of the interstitial fluid. We'll talk more about that later. Okay, the blood also restricts fluid loss. So if there was an injury and you were losing fluid out of your blood vessels, your blood has a special ability to actually create its own blocks to stop that fluid loss. We'll talk more about that later. And then it also helps us defend against toxins and pathogens because there are things called antibodies in our blood. There are things called lymphocytes in our blood, and there are other white blood cells in our blood that help to keep our bodies healthy. And then it also will help to stabilize our body temperature and even regulate our body temperature. For example, your body could send the blood to the surface of your skin if it needed to remove more heat, or it could not send blood to the surface of the skin if it needed to retain heat. One of the ways that our bodies engage in thermal regulation. Okay, we're going to jump into just kind of some overviews, or an overview of the characteristics of blood. For one, it's got a temperature of about 38 degrees Celsius. This is a little bit warmer than your body temperature. The body temperature is about 37 degrees Celsius, so it's a little warmer than that. It also has a variable color to it. It's bright red sometimes. Other times it's more of a dull maroon color, or even kind of like a purplish bluish color. And this just depends on how saturated this protein called hemoglobin is inside the blood, and we'll talk more about that later. It's slightly viscous. This word viscous means kind of thick. So something that with a high viscosity would be very, very thick, almost thinking, like molasses or like a very slow-moving honey or sticky kind of substance. Something that is not viscous would be very thin and would flow well. Well, blood is slightly viscous, but it also does flow really well. It's not super thick. The viscosity of blood doesn't usually change. It can at times, but for the most part, it remains pretty constant. Okay, and then its pH is slightly alkaline. So remember, acidic pHs would be less than 7. Basic pHs would be above 7. This is slightly basic or alkaline, maintaining this pretty narrow range. Now this is an important range for the blood to maintain. If that pH gets too far acidic or too far basic, it can actually start to shut down some things in the body. So the body will work very hard to maintain that pH. We'll talk a little bit more about how it's done later. Okay, the composition of blood. Now, this is what we will refer to as whole blood. Up to this point, I've just been using the word blood, but the word blood can mean a couple of different things. We need to know if we're talking about whole blood, or if we're talking about just red blood cells or white blood cells, and so forth. So in this example, in this slide, we're now talking about whole blood. What whole blood is all of the different components of blood. There's a liquid part to blood, and there's also a cellular part to blood. The liquid part of blood is called the plasma. By the way, this cartoon that we're seeing here is illustrating if we were to take blood and spin it down with a centrifuge, which is a process that's often done in different clinical settings, that the force of that centrifuge would separate out the fluid parts from the cellular parts. And so that's kind of shown here in this cartoon. The fluid part would be this spot. Then there's also what's called the hematocrit down at the bottom. This is made out of red blood cells. And then there's this other last little part here, which is this white layer, which is called, kind of a funny name, the buffy coat. And that is where we would find the white blood cells. So 3 different parts to whole blood: the plasma, the white blood cells, and the red blood cells. And the percentage of the red blood cells to the whole volume is called this word, which you maybe recognize, hematocrit. This is a very important number for clinicians to know if they're studying the well-being of a person's blood. Hematocrit will generally be in a range of percentiles. So if a person was had female anatomy, it'd be their percentage of red blood cells to whole blood would be 37 to 47% volume. So if you took the whole volume of a sample of blood and you wanted to know how much of that blood is actually red blood cells, it'd be 37 to 47% in a person with female anatomy. In a person with male anatomy, that range is a little bit different for a couple of reasons, but we won't talk about those reasons. It's 42 to 52% is the range. If there's an abnormally low hematocrit or an abnormally high hematocrit, that can suggest that there is a problem. These have different names to them. If somebody was to take a person's blood and spin it down and saw that the percentage of the red blood cells to the whole was depressed, we would call that anemia. Meaning anemia means an inability of the body to carry sufficient oxygen. It's usually either because of there's too few red blood cells, or if there are enough red blood cells, there's not enough iron. There's a lot of things that can cause anemia. We'll talk more about that later. Then there's also, if there's an elevated hematocrit, meaning there's far too many red blood cells, that's called also a polycythemia, and both are less than ideal. Now, if we were to look at the plasma and kind of see what kind of things are to be found in it, this is kind of the breakdown. A majority of it's just water. Then there are what we call plasma proteins. There's like basically three main categories of these plasma proteins. There's what we call albumins. And these are the smallest of all of the proteins, and they are also the most abundant. They have a couple of different effects. One of the things that they do is they help you retain fluid. We'll talk more about this in the cardiovascular chapter, but basically water always wants to go to places where there are more particles or there's a higher tonicity is a word you could use, or osmolarity. And albumin doesn't usually leave the blood, it stays in the blood, and so because of that reason there's always kind of a higher osmolarity inside of the blood than there is outside, so it draws water in to try and equilibrate. Albumins also transport stuff, so they're kind of like a taxi cab for lipids and hormones and ions such as calcium. Okay, so they're pretty important. Most abundant and also smallest. There's also a class of plasma proteins called the globulins. There's two different kinds of globulins. There's what are known as immunoglobulins, and this big long word is probably better recognized by you as its more informal name, which is antibodies. So you probably have heard of antibodies. They're things that help clump pathogens together so your body can get rid of them easier. They also prevent you from getting sick through a variety of other ways, but they're called immunoglobulins, and they are in the blood. And then there are what's known as transport globulins. And as the name suggests, these basically just help you your body transport things such as iron or other metals like copper. They'll also help transport fat, lipids, or things like free hemoglobin, which should be packaged up in a red blood cell, but if it's not, it could be transported through a globulin. And then this is the least abundant of all of the plasma proteins, this thing called fibrinogen. And fibrinogen is a pretty cool substance. We'll talk about this also later, but in essence what happens is fibrinogen is dissolved. It's a fluid type protein, and what can happen is it can undergo this big long cascade and eventually get turned or utilized to make this thing called fibrin. And fibrin is insoluble, meaning it precipitates out of solution. It's no longer dissolved, it's no longer fluid, it's now a structural little particle suspended in the fluid of plasma. And the fibrin is going to be kind of the structural patch that gets used to make a blood clot. So that comes from fibrinogen. We'll talk about this whole process in the next lecture when we talk about blood clotting. Those are the three plasma proteins. There's also regulatory proteins. Examples of what we mean by this would be different kinds of enzymes or hormones that are doing stuff like such as insulin or glucagon to regulatory proteins that monitor your or maintain your blood sugar levels. This is a very diverse category, but we won't go into a lot of specific examples here. But we'll become well acquainted with some of these throughout the class this semester. And then other kinds of solutes. So examples would be like electrolytes, potassium ions, chloride ions, calcium ions, sodium ions, vitamins, and amino acids or carbohydrates, things like that. That's the plasma. The formed elements is what we would call the buffy coat plus the hematocrit. All of the cellular contents are the formed elements, and this is the breakdown. Almost all of them are red blood cells. The fancy word for a red blood cell is this one here, which you'll become well acquainted with throughout this lecture, erythrocytes. The white blood cells are given the fancy name leukocytes, and then there's these things called platelets. Leukocytes are the white blood cells, and they're only less than 1%, so by far you can see the majority is red blood cells. Of the less than 1%, there's 5 different kinds of leukocytes. We'll present those later in this lecture. And then there's these things called platelets. Platelets are cool because remember I said one of the plasma proteins was this thing called fibrinogen, and fibrinogen gets used to make this fiber known as fibrin. Well, if there was a blood clot, let's say, here's a blood vessel and it was cut, these little fibrin fibers would kind of anchor into the site of the cut, and then these little platelets, which are these little tiny fragments of a cell, would actually kind of get shoved in there to be the glue that glues all of those fibrin strands together. Okay, platelets are interesting because they're actually cellular fragments. They're not a cell itself. They're from a big, huge cell called a megakaryocyte, and the megakaryocyte will actually shed its cytoplasm in these little packets, which are then going to be found in the blood, known as platelets. And those are also less than 1%. This is a really great exhaustive table that summarizes all of the different parts of blood, and this would be one to kind of star in your notes and work through. You can see this is what we had all already kind of mentioned. There is plasma, and then there's the formed elements. The plasma is made out of water, plasma proteins, regulatory proteins, and solutes. The formed elements are composed of erythrocytes, white blood cells, and platelets. And then there's also some additional information about these. Like, for example, here's the 3 different kinds of plasma proteins. They talk a little bit about where they're made and kind of what they do. So if I were you, I would probably maybe take a screenshot of this and put it on a PowerPoint presentation and just kind of block these off and make kind of your own flashcard and see if you can kind of master this table. I'm not particularly concerned that you know like every single percentile, but otherwise you should know most of the other stuff presented on this table. Some of the disorders that could affect the plasma, we'll talk more about the disorders of the formed elements later, but the plasma disorders, a general disorder known as hyperproteinemia, and this basically just means an elevated protein level in the plasma. Another one would be called hypoproteinemia, which is just the opposite. It's a depressed protein level in the plasma. And the causes of these can be varied. We won't go into the specific causes, but there is a certain type of hypoproteinemia that I do want to make mention of. It's called kwashiorkor. And kwashiorkor, the word comes from the name of this disorder comes from the language of Ghana, and it's translated roughly something as what happens to an infant once it's weaned, or what happens to an infant once it has another sibling. And in essence, what happens is babies, if they are weaned from breast milk, which contains amino acids, so things that are used to make protein, and instead the infant is no longer fed breast milk because there's another baby that's requiring the breast milk, then that infant is instead given a high carbohydrate diet. it can lead to a severe deficiency in protein in the infant. We said that one of the big proteins is this thing called albumin, and albumin helps to retain fluid in the blood. And so if there's a deficiency in albumin, that fluid will no longer stay in the blood, and it is going to pool in some of the tissues of the body. So this is actually a picture of this disorder, and this always, every time I see this, this just breaks my heart that there are small children that are suffering of this disorder. But if you look, you'll see that there is a fluid buildup kind of in the abdomen, and then there's also fluid buildup kind of in the ankles and the feet. Those are usually some of the places where fluid will stay in the tissues if there's not a strong drive for it to return into the bloodstream. This will all make more sense when we talk about the cardiovascular system and how fluid leaves the blood vessels and then it returns to the blood vessels. You also know that there's very thin arms and legs, again due to a protein deficiency. All right, the next thing we have on the docket is this process known as hemopoiesis. This is also sometimes called hematopoiesis. They're both the same thing. And this word is so awesome that I think it needs like a subtitle to it. So I'm titling this Hemopoiesis, Rise of the Formed Elements. This is a cool process which starts with what's known as a multipotent hematopoietic stem cell. Say that 10 times fast. Stem cells are really cool because they are cells that can differentiate into other cells. You know, most often in the body, once a cell becomes the cell that it's going to be, whether it be a bone cell or a muscle cell or a nerve cell, it will stay that kind of cell for its life. But a stem cell is not what we would say differentiated yet, meaning it hasn't committed to being a certain kind of cell type yet, and so it still has the power to turn into any cell, which is really cool. There are also what are known as pluripotent stem cells. Those are the most undifferentiated. They can turn into literally any cell in the body. A multipotent hematopoietic stem cell is limited to just turning into different types of blood cells. Okay, so we're starting off with this cell, this stem cell that can turn into any kind of blood cell, and it will divide, and one of the daughter cells of that division will actually stick around, and it will be used to replenish the next round of dividing, because you can imagine if this didn't happen, and these cells just went and both became differentiated blood cells, then we would quickly run out of our stem cell population. But because this sticks around, the stem cell population is maintained, which is good for us. It means we can always make blood. Then the one that didn't stick around is committed now to differentiating into a certain type of blood cell. And it's got two paths it can go down. One is called the lymphoid stem cell lineage. The other one's called the myeloid stem cell lineage. The lymphoid stem cell lineage will give rise to these cells which we call lymphocytes, which is what we call this lymphoid. And then this other type of lymphocyte, which is called a natural killer cell, which I know that has a pretty awesome name, doesn't it? We'll talk a lot about these when we get to the immune system chapter. Of the myeloid stem cell, then it's everything else. And here's where we're going to first start to see just how complex the formed elements are. Here's the erythrocytes. These are the red blood cells. And then these are all of the white blood cells. These are also white blood cells. Okay, and now you can see the different kinds. We have basophils, neutrophils, eosinophils, monocytes, and lymphocytes. For now, you can just group these all as one type of cell, which we would call a lymphocyte. When we get to the immune system chapter, we'll break them down into the three subtypes. And then the platelets, which come from this big gnarly cell, which is called the megakaryocyte. For now, it's important that you just kind of keep in mind these, and then in part 2 of this chapter, we'll then go in and we'll talk specifically about what each one of these types of cells does. Now, what's kind of interesting is that even though the stem cell will turn into either a myeloid stem cell or lymphoid stem cell, some of these cells the production of them, it can be governed by different hormones. So, for example, there's a hormone known erythropoietin, and if that's released by the body, it will drive this particular process right here of turning a myeloid stem cell into the precursor cell that gets made into a red blood cell. So all that to say, if the body needs more red blood cells because of a condition known as hypoxia, which basically means just low oxygen in the body, this could be due to a variety of reasons. One, anemia, meaning there's not enough red blood cells to carry the oxygen. Two, there's not a lot of blood flow, specifically to the kidney. This is kind of a weird one. We'll talk a little bit more about that later. The kidney is kind of the place where this hormone is made, so that's why blood flow to the kidney is important. Low oxygen, let's say we're up on Mount Everest and there's a low altitude or high altitude, there's not as much oxygen around, that would cause low oxygen. And then respiratory damage, even if there's enough blood and there's plenty of oxygen in the atmosphere, it could be that there's respiratory damage so that that oxygen is not actually getting from the atmosphere into our bodies. Either of those conditions would lead to low oxygen in our body, and the kidney will detect that and release this stuff called erythropoietin, which is going to drive the production of red blood cells. So that's what our bodies are doing all the time when we become low on red blood cells. In contrast, there's another regulatory hormone called a variety of them called the colony stimulating factors that will, when that's released, it will produce or it will encourage the myeloid stem cells to divide into these cells, which will give rise to these. Okay. Now this is kind of a big convoluted flow chart. I'm not requiring you that you know all of these precursor cells, but you should know that there's two lineages: the myeloid stem cell lineage, the lymphoid stem cell lineage, and then you should know what is ultimately made. These ones, and then for this chapter, just know that these are lymphocytes. Don't need to know the specific ones quite yet. And then the two hormones, erythropoietin and colony stimulating factor. Erythropoietin for red blood cells, colony stimulating factor for white blood cells. There's also some that are used to encourage the production of platelets. There's also some other ones used to drive production of white blood cells, but we'll just leave it at those two regulatory factors for this class. All right, here's a time for you to just take a moment to review. I've got a couple questions here. You can go ahead and pause the video. You should at this point be able to answer these questions. See if you can do it from memory, and if not, go back and look at what we had talked about. I would say when you're preparing for the first exam, it would be a really good idea to go back to these review questions and make sure that you can answer them all to the extent that we had talked about in lecture. Okay, the next big part of this chapter is all about specifically red blood cells. Again, the name for a red blood cell is this, erythrocyte. We're going to start by talking about some of their characteristics or their features. Red blood cells are pretty cool. There's a lot of them in your body. In fact, about 1/3 of all the cells in your body are red blood cells, so that's pretty fascinating. This is maybe even more fascinating. If you were to take 1 microliter of blood, which it would be basically 1 cubic millimeter, maybe if you know about how long a millimeter is, you can kind of conceptualize how much 1 microliter is. Well, that will contain 4.2 to 5.5 million red blood cells. Now, if you were to take a cross-section of a red blood cell, you know, this is showing a red blood cells on a scanning electron micrograph. So if you were to take one of those and take a cross-section of it, it has kind of this shape to it. We call this shape a biconcave disc. And the biconcave disc is so named because we have on one face of the red blood cell a concave kind of indentation, and on the other side of the blood cell we have kind of a concave indentation. Remember, if this was a plane, this would be a concave shape, and this would be a convex bend. So we have two concave bends, so it's biconcave. The purpose of this is to help increase the surface area that can participate in gas exchange, or mostly just gas exchange. So, imagine if this was just a disc and the wall did not bend in, but it instead just went straight across like this. That would be, a certain distance, right? Well, if we bend that distance down, it actually makes the distance a little bit longer. So there's more surface area, even the same size cell, there's more surface area for oxygen or CO2 to get into and out of that cell. And when we're talking about gas exchange, which is what red blood cells are kind of champions of, uh, surface area is key because you have to get rid of as much gas as fast as possible because those red blood cells might only, swing by the lungs for a couple of seconds, and in that couple of seconds they have to exchange all of their gases. Okay, just the size of these cells, they're about 7 to 8 microns thick. Some of the smallest capillaries in the body are only 5 microns thick. And so you can see that this red blood cell would have to fit through a tube that is less wide than the actual cell. So another property of these red blood cells is that they're flexible. They can squeeze into very small spaces. Okay, some of the other things that I should mention here is that they are anucleate, meaning no nucleus. These are one of the, not many cells in your body don't have a nucleus. In all of the red blood cells, which I guess there's a third of all of the cells in your body, so I guess a lot of cells in your body don't have a nucleus. But they don't have a nucleus. The purpose for this is that the job of a red blood cell is really to transport oxygen, in essence. Okay, it does other things, but that's one of its huge jobs. And so it's kind of like you got to strip that cell full of all the unnecessary equipment and just fill it chock full of stuff that it needs to actually do its job that it's famous for. So most of the organelles have been stripped out of this cell. The nucleus has been stripped out of this cell. Those take up a lot of room, and instead it has been packed full of this protein called hemoglobin, and hemoglobin will carry the oxygen. Now, because of this, a red blood cell is going to have a short shelf life. A cell requires a nucleus to maintain itself. The nucleus, again, is what holds all the information required to build proteins, and a cell that can't make protein is a cell that has a limited shelf life. The shelf life of a red blood cell is about 100 to 120 days. Okay, they also lack mitochondria, so they don't really make energy through aerobic respiration like a lot of other cells in our body, like muscle cells. They will actually make energy by taking glucose and breaking it down through glycolysis, and then the pyruvate that is made will be fermented and actually used to make energy. And you can imagine it would be kind of a poor setup if these red blood cells make energy from the very thing that they're carrying. It's like what happens to cookies in my pantry if they're around. I make energy through cookies, and if I have a pantry full of cookies, I'm going to eat all those cookies, okay? If this red blood cell makes energy through oxygen, it's got tons and tons of oxygen, maybe those red blood cells would be a little tempted to sneak into the oxygen jar and use all the oxygen that they're supposed to be giving to other cells. So all that to say, it's good that they don't use oxygen to make energy. Okay, we already mentioned this large surface area to volume ratio. That's the biconcave disc. They're also very flexible. They can get as narrow as 4 micrometers, so they can almost get half of their size. They will form what are called, you got to lean your shoulder in a little bit when you say this French word, the rouleau. All right, give it a try. Lean that shoulder in, rouleau. The French word rouleau really means rolls. And so these can form little stacks. Almost think about it like a can of Pringles and all of those Pringles stacked on top of one another. What you're seeing here is a scanning electron micrograph of a whole bunch of red blood cells all stacked on top of one another. They kind of stick to one another temporarily, and it's not very strong, the stick, but it's enough to get them to stack. This is good because if you have to shove a whole bunch of red blood cells into a tiny little tube, you don't want them, exploring the space and sprawling out. You want to get them tightly packed into a little roll and shove them down the tube. Now there's a really cool video, and I could probably watch this video all day long. I love watching videos of red blood cells go through vessels. But you can kind of see some of that, what we're talking about here. Each one of these is a little red blood cell, and you can see them all kind of, stacking together at times to kind of fit through this very small capillary. This would probably, what you're seeing here, be a capillary. That's the smallest tube that a red blood cell would fit through, the smallest vessel in the cardiovascular system. And that's probably draining into this tube, which is probably a venule, which is one of the smallest veins. All right, let's talk a little bit about hemoglobin. Hemoglobin is essentially what is in a red blood cell. 95% of all of the red blood cell's proteins are hemoglobin. So really a red blood cell is just a big sack of hemoglobin. Okay, there's about 280 million hemoglobin molecules. Each hemoglobin molecule can carry 4 molecules of oxygen. So that means that there's like a billion molecules of oxygen that one red blood cell can carry. Each hemoglobin has on it 4 what are called heme groups. And hemoglobin itself is kind of an interesting protein. It's a quaternary protein, meaning, or it's a globular protein, meaning there's several proteins attached to one another. So there's a protein here, a protein here, a protein here, and a protein here. And there's 4 proteins all stuck together to make one super protein known as hemoglobin. Now, inside of each protein is this special little thing called a heme group. It's basically an atom of iron and surrounded by a bunch of other stuff. Okay, so right in the middle here is an iron. This is an iron. This is an iron. And iron is the thing that is attracted to oxygen. So when a bunch of oxygen molecules enter, I'm drawing these as O2 because that's how oxygen is arranged in the atmosphere. We call it diatomic oxygen, 2 oxygen atoms connected together. When that enters in, those oxygen molecules will go in and they'll bind to the iron that is present on the hemes of each subunit of hemoglobin. So you can kind of think of hemoglobin in essence as being kind of like a taxi cab for oxygen. And it's a taxi cab that has 4 chairs. Each heme is the chair. That's a place where oxygen can sit. Now, this will carry nearly all the oxygen in the blood. 98.5% of all of the oxygen in your blood is bound to the hemes and hemoglobin. But you can also see there's about 1.5% that is not actually bound to hemoglobin. It's just inside the blood, unbound. Now, when an oxygen molecule enters the blood from the air, and then it goes and it binds to a hemoglobin, we no longer call this a molecule of hemoglobin, we call it a molecule of oxyhemoglobin. And oxyhemoglobin is bright red, which is why blood that has oxygen in it is red. This binding of oxygen to the heme, it can actually change the strength with which it binds, but generally it's a pretty weak interaction. That's important because oxygen needs to get on really quick and it needs to get off really quick. If oxygen bound really tightly to the heme, then when this red blood cell passes to some part in the body where it has to deliver the oxygen wouldn't want to go. It would stay stuck onto this heme group. But because it is a weak interaction, it can jump off. Now, there are things that can affect how strong this interaction is. Temperature and pH are some of the good examples, and we'll talk more about that when we get to the respiratory chapter, or the respiratory chapter. Now, one could measure roughly how many chairs are occupied by oxygen. Okay, so if we wanted to know roughly the percentage of all of the hemoglobin molecules in a portion of blood, how many hemes of all of those hemoglobin molecules are occupied by oxygen, that's called the hemoglobin saturation. And you can measure hemoglobin saturation with a tool which is called a pulse oximeter. And a pulse oximeter, what it does is it shines 2 beams of light. One is infrared, so it's not absorbed by the hemoglobin. Another is not infrared, so it is absorbed by the hemoglobin, and the degree to which it's absorbed depends on how saturated this is, because remember, these are different colors. If it's really, really saturated, it's a bright red color. If it's not, it's a different color, and so the shape change of this protein as it becomes more and more saturated will change the degree to which how much light is going to be absorbed by it. So it can run some fancy calculations to figure out just how much light is absorbed by hemoglobin, and that would tell us something about just how saturated it is. And this is a pretty cool device because you can clip it right onto some place where there's a thin kind of window. Usually they use the fingertips because this is where light can pass through that hemoglobin, and then it will detect the light and then give a readout here of not only the oxygen saturation, but also oftentimes the heart rate. Okay, now if there was a low oxygen saturation, what would happen is that there are specialized receptors in your body, and the place that these receptors are found, I always think that's really cool that you could have a receptor in your body that can actually detect how much oxygen is in the blood. I mean, what does that receptor look like? I don't know the mechanism there, but I'm sure it's pretty elaborate how a cell can detect the oxygen concentration in a space. But there are specialized receptors cells that detect the oxygen level of the interstitial fluid in the kidney. Specifically, these cells called fibroblasts that are found in the kidney will detect the level of oxygen that they're encountering in the interstitial fluid. And the kidney is a good place to detect oxygen level because about 20% of your blood, 20 to 25% of your blood is passing through your kidneys right now. Okay, 25% at least of your cardiac output, the amount of blood that's left your heart, will find its way to the kidney. So they're a very highly vascularized organ, good place to detect how much oxygen is in there. Now, we said earlier that this thing, erythropoietin, is released to drive the production of red blood cells. Well, erythropoietin is released by those fibroblasts in the kidney specifically, and if there was a low oxygen saturation, the erythropoietin would be released to create more red blood cells so that more oxygen could be carried and likely, hopefully, increase that amount of oxygen that's being carried in the blood. Okay, another quick note here is that CO2, interestingly enough, also binds to hemoglobin. Except CO2 doesn't bind to the iron like oxygen does. Remember, oxygen goes and binds to the iron of the heme. CO2 instead will bind to the amino acid part, so the globular parts of the protein. And that's going to form a compound which we call carbaminohemoglobin. Now, this kind of begs the question, why use hemoglobin? Why not just have oxygen go into the blood and then come out of the blood? Why does it have to ride on these little chairs? Let's think about, like, if a freeway was full of buses that were carrying lots of people, the freeway would be much less congested than if you were to just let all of those people loose and have them walk down the freeway themselves, right? So imagine a freeway with just a bunch of people walking down it. That would be packed full of people, and it'd be very chaotic, and it'd really block the freeway. Well, in the same way, if you have a blood vessel that just has a lot of oxygen atoms kind of bouncing around, that is congested. It's not as organized. Whereas if you have these little shuttles that all of the oxygen can jump onto, then it just makes it so that there's not so much spread and sprawl of the oxygen molecules, and they also, it's going to play an importance in how oxygen is delivered to tissues, which we'll talk again about that when we get to the respiratory system chapter. There are some organisms that don't use hemoglobin, but for us it greatly increases the efficiency of how much oxygen our blood can carry. Okay, a disorder that involves the hemoglobin protein, there's a mutation that can happen, and I think it's if I recall, it's a point mutation, meaning it's one little base that gets mutated. Don't quote me on that. I can't recall off the top of my head. But in either case, it leads to a misshaped folding of the hemoglobin. That actually will manifest itself in a cell that has an abnormal shape to it. So, blood cells have a lot of hemoglobin in it. So if the hemoglobin is misfolded and the red blood cell has lots of hemoglobin that's misfolded, then the actual cell itself will become misfolded. And in this disorder known as sickle cell anemia, the red blood cells, which normally look like a biconcave disc, that actually kind of flattens out and gets these points on it, and it looks kind of like a sickle. A sickle is a tool used by farmers a long time ago, or in farmers in less probably developed areas, and it's just like a kind of like a sharp blade. But in either case, it looks kind of has this shape, so that's why we call it sickle cell anemia. These sickle cells are not as flexible. They clog up in capillaries. They don't form those nice rouleaux, and they just don't carry oxygen as well as normal red blood cells. Okay. Also, these cells can lyse easier, so they can get broken down. Okay, the last thing we're going to talk about here is the life cycle of an erythrocyte. And I always really enjoy this part of the lecture. It's pretty interesting, I find. So how red blood cells are made is they start off, again, with our hematopoietic stem cell, but it happens in the bone marrow. The bone marrow, some of the important places that it's found is in the epiphyses of long bones. So like here you're showing the epiphysis of the humerus, the head is the informal name of epiphysis. You're also seeing the epiphysis of the femur. You also can see it in some of the flat bones. So you can see it kind of in the flat bone here of the pelvis. There's also some bone marrow in the sternum as well. And I always thought that was interesting that red blood cells are made in the bones out of all places. We're tempted to think that bones are just these lifeless hunks of mineral, but they actually are the factory that produce the life-giving substance of blood. Well, in the bone marrow, the stem cell is going to get converted to this cell, which is called a reticulocyte. Inside the marrow, there is a reticular connective tissue. And so that's kind of why we call these reticulocytes. And a reticulocyte will then enter the blood. And shortly thereafter, entering the blood, within about a day or so, they will mature into a red blood cell. The reticulocyte note here does not have a nucleus. So there was a cell called an erythroblast. The erythroblast lost its nucleus to become a reticulocyte. Then the reticulocyte, which is anucleated without a nucleus, went into the bloodstream, and then it got converted into this final shape here, the erythrocyte. Now after that happens, the clock starts ticking. After the nucleus is lost here, the clock starts ticking. We only have about 120 days of that cell to live. So that cell will go into the bloodstream. It will live for 120 days and do its tour. And then after about 120 days, it starts to get worn out. Some cells can die before 120 days. Some cells can last longer than 120 days, but 120 days is the average lifespan of a red blood cell. Now, at the end of those 120 days, the cell will either break apart into fragments, or it will get swallowed up by a phagocyte. And the phagocytes are usually found in the bone marrow, in the liver, and the spleen. The phagocytes are also called macrophages. Okay, so macrophages in the bone marrow, liver, or the spleen, 3 different spots, will be monitoring the blood and looking for worn out, tired red blood cells. Or they'll be trying to scoop up fragments of red blood cells that have already hemolyzed. Hemolyzed is the word used to describe the break of a red blood cell. All right, in either case, what happens is that the macrophage that scoops up the parts of the red blood cell or the whole red blood cell will, in either case, break it down. And then there's a bunch of fates of the different materials used. The hemoglobin, which is 4 proteins, will kind of disassociate into its individual parts. And the protein part, which we call the globin portion, right here, will get broken down into amino acids and other cell stuff. Now, the heme then there's 4 hemes in a hemoglobin, has kind of a special fate. The heme gets split apart into its pieces, the iron atoms, that's the little thing you find in the middle, and then the rest of the heme. And then these have more of a special fate to them. The iron ions are valuable, and the body can reuse those to make more red blood cells. But the caveat is that iron, as well as other heavy metals, can't just ride around in the blood. That's not good. Okay. Usually heavy metals have to have a chaperone. And so the iron is bound to this thing called transferrin, which is going to hold on to it and guide it through its journey back to the bone marrow, where it can be recycled. The heme group has a more of a convoluted journey. It's going to get broken down in this thing called biliverdin, and biliverdin will be in the blood. Biliverdin is sometimes transported with albumin through the blood, and it's going to make its way to the liver. Okay, so the biliverdin will make its way to the liver. Inside the liver, and actually the biliverdin will probably get broken down into this stuff called bilirubin, and then the bilirubin is the thing that makes its way to the liver. Also, iron that was not recycled to the bone marrow can also be stored in the liver as kind of a bank for when iron is needed. So if there's a surplus of iron, iron gets loaded not onto transferrin, but it gets loaded onto this thing called ferritin, and ferritin is found inside the cells of the liver. It's also found in other cells, but it's in the cells of the liver. And so the liver is kind of a nice storage bank of iron, and then if need be, it can get loaded back onto transferrin and loaded back over to the bone marrow to make more red blood cells. So transferrin is what carries iron in the blood. Ferritin is what carries it and holds on to it inside of a cell. Okay, now this is what I find to be interesting. I mean, it's all interesting, but this is extraordinarily interesting. Bilirubin inside the liver gets converted to this stuff called bile. Okay, bile is used. It's released out of the liver into the small intestine, where it will help you absorb fats. Okay, so now we've got bile in the small intestine. It's helped us absorb fats. Well, it will travel down the small intestine, then it will reach its way to the large intestine. And in the large intestine, bacteria will take bile, any bile that remains, and they will convert it to this substance which is called urobilinogen. Urobilinogen could get converted into stercobilin, and stercobilin is brown. Urobilinogen could stay, or it could be excreted in the feces, or it could be reabsorbed in the bloodstream where it becomes urobilin. And then if it gets converted to urobilin because it's reabsorbed out of the large intestine back into the bloodstream, it will eventually then be excreted in the urine as urobilin. Okay. So here's what I'm trying to point out. Urobilin, when it's oxidized, it reacts with air, it turns yellow. So here we have a brown colored substance, a yellow colored substance. The brown color that is stercobilin, this is what makes your poop brown. Okay? And the yellow color in urine, that's from the urobilin. So here's what the joke that I always tell. Anytime you go to the bathroom and you're looking at the toilet and you see your yellow pee or your brown poop, go ahead and just give yourself a salute. Salute the red blood cells that have done their 120-day tour and given their life to supply all of your cells with oxygen. I just find it fascinating that the color of our poop and the color of our pee is from red blood cells. That's the last thing I would have thought to have been the thing that makes our poop and pee have its color. But it isn't the case. And that's actually a good point to bring up, because if there's some abnormality in the color of one's feces or the color of one's urine, it would suggest perhaps some abnormality with this whole process. Either how many red blood cells are being made and released, or how well the liver is converting all of these things, so on and so forth. Now, there's a lot that I've covered here. You don't have to know the specifics of all of the stuff I've drawn here. I do want you to kind of have a good understanding of what's shown in the figure here. I also think it's kind of interesting that you know that stercobilins and urobilins are brown and yellow. Stercobilins are excreted in the feces, urobilins are excreted in the urine, and these ultimately come from the heme group of red blood cells. So, interesting stuff there. And oftentimes when they're assessing liver function, as well as other types of blood stuff, they will do a test, and you might look at your lab results and see things like bilirubin and things like ferritin and transferrin and stercobilin or urobilin. Because if there is a buildup of bilirubin, that means that the bilirubin is not being converted to bile, and it's not being released out of the body. So maybe there's a blockage in the gallbladder, or perhaps there is something wrong in the liver where it's not being converted into bile. So that would be kind of interesting to know. If there's low levels of ferritin and transferrin, that likely means that the body's not able to reuse its iron, so there could be a lower amount of red blood cells or a depressed hematocrit. So all sorts of things that could be told by examining the level of these different substances in the blood. Okay, so that leads us to erythrocyte disorders. One of them is hyperbilirubinemia. And believe it or not, I actually went to high school with a kid whose name was Billy Rubin. I don't know if his parents were aware of what bilirubin is, but now looking back on it, I think it's kind of funny. Hyperbilirubinemia, oh, it's hard to say, is excess bilirubin. Okay, so there's a lot of more of this around, and what that can lead to is an overproduction of bile. And bile is a way of being able to sneak its way into tissues. So if it's not, if there's too much of it made and it surpasses the amount that is drained into the small intestine, well, then that bile is going everywhere. Okay, if it's not drained through the small intestine out of the body, it will accumulate and it will get into the bloodstream, and out of the bloodstream it will go everywhere. It will diffuse into the tissues, and bile is a yellow color to it, and so when it does diffuse into the tissues, it makes the tissues yellow. This is a condition known as jaundice. One of the places that is probably most evident is in the whites of the eye. We notice it probably fastest here because this is, as we all know, is supposed to be mostly a white color, and so the yellow colorization is most easily visible, but there would be yellowing occurring in all of the different tissues. This would suggest perhaps that there is a blockage which is not allowing the bile to leave, or perhaps there is some condition which is leading to an over-destruction of red blood cells. Too many red blood cells are being destroyed, which is why the amount that bile is produced is elevated, because there's much more bilirubin made from this process than what would normally occur. So a couple different reasons this could happen. This also happens in infants occasionally, especially if they're premature infants, because the liver isn't quite fully functional yet, so they might have jaundice, and if that's the case, they will sometimes subject them to this blue light, and that blue light is interesting in that it actually strikes the bile and breaks it down. Okay, another big one is anemia, and we should pause to talk some time about this, because there's lots to say here about anemia. Anemia is a fairly common thing that happens, and there's a lot of different reasons why. Basically, anemia, the definition of it would be the inability of one to carry oxygen in blood, or there's not enough oxygen in the blood to meet the metabolic needs of the body. This could be for a variety of reasons. It could be because of blood loss. If you lose a lot of red blood cells, then you will not have, I guess, carry oxygen in the blood to meet metabolic demands. Likely that during anemia, likely it's still possible to carry oxygen, but just not enough. Okay, if there's an increase in red blood cell destruction because maybe of a certain virus or pathogen which is destroying the red blood cells, or an example of this would be like sickle cell anemia, or another reason because there's a decrease in red blood cell production. This could be for a couple of reasons. One, iron deficiency. Or two, this thing known as pernicious. Sorry, this is an E. I should actually just not write so poorly here so you know what I'm actually trying to write. Pernicious anemia. Pernicious anemia is a name of a disorder in which this vitamin known as B12 is deficient. B12 is a vitamin that's helpful for the process of red blood cell production, and so if B12 is not around, then the erythrocytes aren't produced as they ought to be. There's a decrease in red blood cell production. Also, if there's not enough iron around, the red blood cells won't be produced. Okay, so a couple different causes here for anemia. Now, the iron deficiency one, there's kind of an interesting photo here showing the difference. I'll see if you can guess which of these is normal red blood cells and which is not. The normal one is over here, and abnormal would be over here. So if you look at this slide, you see that the red blood cells are have a good colorization to them. They have kind of a nice purple to them. Here you'll see a little bit of purple on the outer rim, but there's a lot of pale color in the middle, meaning there's just not a lot of substance there in the middle, that the light is passing through the cell. And this is because this is a picture of somebody with iron deficiency. The red blood cells are a little bit more pale. All right, let's take a little time to review here. I've got a couple questions for you to consider. List some of the characteristics of erythrocytes. Describe the structure and the function of hemoglobin. Draw out, this is going to be a challenging one, so spend some time with this one. Draw out the life cycle of an erythrocyte, and then also describe it. In your description, you should be talking, making mention of some of these things here that I've listed. Describe some of the erythrocyte disorders, the ones that I've just mentioned. And then lastly, I want you to go and research and try and explain this thing called blood doping. I didn't talk about it here, but this is something that is an issue in sporting competitions for some athletes. So research that and see if you can come up with some information and explain what blood doping is. We're going to stop here, and when we get back, we're going to venture on to talk about the white blood cells.